everyone, welcome back to the channel MLT Lectures. In today's lecture, we will discuss about catalase test, a common biochemical reaction done to differentiate between Staphylococcus species and Streptococcus species. Before moving towards the video, don't forget to subscribe the channel. Now let us see why catalase test is done. It is used to detect the presence of an enzyme, catalase, in an bacteria to differentiate between Staphylococcus and Streptococcus species. It is also used to differentiate between aerobic and obligate anaerobic bacteria. Now let's see the principle. Here we can see that catalase is an enzyme that converts hydrogen peroxide into water and oxygen. Here in reaction we see H2O2, that is hydrogen peroxide, that is acted upon by catalase enzyme and break down into H2O, water, and O2, oxygen. The bacteria that contain this catalase enzyme are usually aerobe, which need oxygen, or facultative anaerobes, which can live with or without oxygen. Now, doing this test in the laboratory, we need 3% H2O2, a clean dry slide, inoculating loop or wooden stick, tubes. To determine the hydrogen peroxide reagent is working, we must do quality control by using positive and negative control animals. For positive control, we use Staphylococcus species, and for negative control, we use Streptococcus species. To determine catalase test, we have two methods. One, slide method. Two, tube method. First, let us discuss about slide method. Transfer a small amount of bacterial colony to the surface of a clean dry slide using loop or a wooden stick. Then, place a drop of 3% H2O2 hydrogen peroxide on the slide and mix. A positive result is the rapid evolution of oxygen within 5 to 10 seconds, as evidenced by bubbling. The 5 to 10 second time should be observed very carefully. A negative result is no bubble. After the test is finished, dispose of your slide in a biohazard glass disposal. Here in figure we can see positive and negative reaction. In positive slide we can see bubble formation or effervescence. In negative slide no bubbles are formed. Moving to test by tube method. Add 4 to 5 drops of 3% hydrogen peroxide to a test tube. Using a wooden applicator stick, collect a small amount of organism from a well-isolated 18 to 24 hours colony and place into the tube. Don't pick up any agar, especially blood agar, during this test. Place the tube against a dark background and observe immediate bubble formation at the end of the wooden applicator. In this figure, catalase positive tube contains a bubble at the end of wooden stick. In catalase negative, no bubble formation. Result: catalase positive reaction evident by immediate effervescence, that means bubble formation. Catalase negative reaction, no effervescence. Thank you. Hope you like the content. Please like, share and subscribe the channel.